So we'll start the class. Okay, let's start the class. In the last class, what we have discussed is we have talked about the first phase of the cell cycle, that is we have talked about interphase, right? So what we remember, the entire cell cycle. Okay, first let's say about the cell cycle. What is cell cycle? It is nothing but a sequence of events that undergoes or that a cell undergoes prior to division and together with division, right? A cell has to form two daughter cells, and before division ki changes hoy, everything comprised of the cell cycle. Broadly divided into two phases, that is the interphase and the dividing phase. When we talk about the interphase, it is also called as the preparatory phase, and it is divided into three stages, that is G1, S and G2. Important points about G1, S and G2 in the form of a chart we have already discussed, and we have also discussed something called as a G0 phase, where the cells remain there either permanently or temporarily, and if they are on temporary basis, they start dividing whenever the necessity arises, right? Today what we are going to discuss is, I think last class you have already written the point, no? The topic for today is why does a cell divide, right? As far as this portion is concerned, it's not very important from examination point of view. Why does a cell divide? Ketiyao question aha nai, right? This is something extra that we say since we are talking about cell division. So eta bostu janibole is a cell to divide kiyo kore. Examination point of view pra ekku aha nai, and we will be not discussing the entire contents as to why does a cell divide. Various scientists from time to time have given different opinions regarding why does a cell divide, right? But we will be talking about just two of them. So likhi lom first, because of why does a cell divide. Kernplasm, PLASM, Kernplasm theory or Karyoplasmic Index. Index par ratio is equal. Okay, so we will be talking about two opinions. First is a cell divide because there something happens between the surface of the cell and the volume of the cell, and second, according to some, a cell divides because something happens in the nucleus, right? Since we have a word called as kern or karyoplasmic or karyokinesis, whenever we hear this word, we know that we are talking about the nucleus, right? So first we'll come to the first one, surface volume ratio. Ki koisile je eta kaam kora likhilba. Okay, heading koribason surface volume ratio. Extra bole likhi thoba, huh? You don't have to read so much. Okay, cell eta thakibo, cellor bhitor ki thakibo, organelles thakibo, and you have the cytoplasm also inside. What does the surface volume ratio tells us? It tells us that, so jiman bilak contents ase cell etar bhitor, it's dependent on what? Dependent on the nucleus, and it's also dependent on the cell membrane also, karon cell membrane opra tahate ki bur pabo, whatever they want in and out hoi thakibo. So what does this tells us? It tells us that the cytoplasm content inside a cell keeps on increasing because the cells grow. Eta cell to grow koriboi lagibo, so the cytoplasm content keeps on growing. Plus we have already talked about duplication of the cell organelles, right? Interphase or ki hoy, maximum of the cell organelles duplicate, right? So as a result, the content inside the cell increases as compared to its surface. Surface or pra tahat ba surface or uporot organelles bilak ba cytoplasm bilak ki hoy dependent hoy ase. Jete inside portion besi hoy jai as compared to the surface, as compared to the cell membrane, then the cell is bound to divide. Ratio to ifal hoy jai na, because you have a particular content that a cell should have. Eta cell division, prior to division, contents bilakor eta amount thakibo. Jeti amount to barhi jai, what they will do? They are bound to divide. So this is all about, in one line, that is the meaning of this sentence, okay? And the other one is, okay, we'll talk about it later. Eto likhilwa, likhibo okman basically lagibo, kinu meaning hetue. Okay, eto to buji paiso no? It's because the cells grow much faster, and so the membrane becomes incapable of supplying the nutrients that they require, and as a result the cell is forced to divide, right? Tahator ratio to cell to basi fast grow kore, so membrane it's no longer capable of supporting that cell, and so the cell divides. That's the first one. Second is something called as Kernplasm. Yate ki koise, this is in relation with the nucleus. What it says is, when we talk about a cell, obviously everything is controlled by the nucleus, because nucleus is called as the brain of the cell. Hetu to ami janu, right? So nucleus e ki kore, gutei bilakok control kore, but how far will it be capable of controlling? Because we know that the cytoplasm will keep on growing and organelles will also keep on duplicating. So eta nucleus e jiman kini control koribo pare, jedi tar contents to barhi goi thake, tetiale at a time what happens, the nucleus become inefficient to control the newly formed organelles, and so the cell is bound to divide. Etai ki koise, because of the surface contents besi hoise, surface incapable hoise, right? Second ki koise, কন্টেন্টস বেছি হৈ গৈছে নিউক্লিয়াছে কন্ট্ৰোল কৰিব পৰা নাই নিউক্লিয়াছে কন্ট্ৰোল কৰিব পৰা এটা কেপাচিটি আছে ৰাইট সো অৰ্গানিজ মেবি ইফ সাপোজ ইফ আই সে নট আ ভেৰি সাইন্টিফিক স্টেটমেন্ট তথাপি মই যদি কৈছো ইফ ইটস কেপেবল অফ কন্ট্ৰোলিং জাস্ট 2 মাইটোকন্ড্ৰিয়া পাৰ সেল যদি অল অফ আ সাডেন 4টা হৈ যায় নিউক্লিয়াছৰ ওপৰত প্ৰেছাৰ পৰিব সো হোৱাট হ্যাপেন্স ইজ দ্য সেল ফোৰ্স টু ডিভাইড right these are all hypothetical not a very authentic statement that gives us a reason why does a cell divide right both scientists have beleg koise but we will be talking about this one and this one why contents besi hoi jay nucleus e control koribo noare he karone divide kore or contents besi hoi jay membrane e support koribo noare he karone divide hoi jay clear
nucleus e control koribo noare that means in what functions nucleus
synthesis of proteins right synthesis mainly to nucleus of proteins no RNA hok kaheri hok or ki hobo genetic material synthesize about different types of proteins they will synthesize our proteins fully call it it's a building block of the cell so ultimately it is controlled by the nucleus itself right okay So why does a cell divide? A cell divide because of surface volume ratio, or it divides because of Karyoplasmic Index. Whenever we talk about Karyoplasmic Index, we are talking about the nucleus. What is the point that we are studying? We are studying the volume. Right? Kihor volume, eta cellar bhitor ki thakibo, nucleus thakibo or cytoplasm thakibo, duitar volume compare kori pele ultimately we will have an index or a measurement that is called as Ki. Talking about the Ki, or the Ki tells us it's time for the cell to divide. So cell division kihor upor depend kore, Karyoplasmic Index upor depend kore. What is Karyoplasmic Index? Is nothing but the ratio. Ratio kihor between the size of the nucleus and the size of the cytoplasm. Size boli kore, size of cytoplasm is not the term that we use. Generally we use the volume of the cytoplasm, right? Size of cytoplasm boli to eko kotha nai. So ei duta bostu compare korisu, ki compare korisu, cell to bitor nucleus or volume or cytoplasm or volume, right? So nucleus to, as we have already discussed, no, cytoplasm jodi basically grow kori thake, tetihole this volume will get disrupted, right? Ultimately the cell would be forced to divide. So these are the two different opinions that is given to us by the different scientists. Now from examination point of view, what it has been asked, eblak to eko hudha nai, ketiaba ei bostu to upor hudibo pare, just this one, right? Karyoplasmic index is equal to volume of cytoplasm by nucleus, ne nucleus by cytoplasm. Okol ei bostu to monot rakhile hobo. Baki in details, what is the ratio, what does the ratio tells us, eko hudha nai question. Ketiaba etu ahibo pare. Extra poli likhi thoi dia side because you might forget. Okay, so up till here what we have studied is we have talked about the preparation phase and why does the cell divide. Next is we have to talk about the M phase or the dividing phase, and when we talk about the dividing phase, either we have to talk about mitosis or we have to talk about meiosis. So what is the difference between both of them and what is the similarity? Mitosis is the process of division, meiosis is also a process of division where the mother cell divides and forms two daughter cells. But as far as the number of daughter cells that is formed is concerned, mitosis or eta cell divide kori pelai keita daughter cells form kore duta. And if you are talking about meiosis, then obviously the product is 4. That's one difference. Second difference is, second similarity is the contents are equally distributed and the contents are not equally distributed. Contents boli kole we are talking about the genetic material, the contents of the nucleus, the contents of the cytoplasm, and specifically we are talking about the chromosomes, right? So chromosomes divided equally between the daughter cells, right? Parent cell or jiman chromosomes asile 46 asile. Suppose if the parent cell has 46 and if the daughter cells also has 46, after division, then that type of a division would be called as equational division, and that is what is called as mitosis. Kintu if we see that parent cell or jiman asile 46 asile, daughter cell or jiri 23 thake, it's completely a reduction in the chromosome number, and that process would be called as ki reduction division, and that is what is called as meiosis. Talking about mitosis and meiosis, where can you see mitosis and where can you see meiosis? If you are talking about body cells bilakot ki dekha pao mitosis. If you are talking about reproductive cells, right, or germ cells, tete ami tat ki dekha pao meiosis, right? So next phase we have to talk about mitosis and meiosis in details. As far as mitosis and meiosis, the common thing is the parent cell will always divide. Whenever a parent cell divide or whenever a cell divide, there are two processes that are involved, the division of the nucleus and the division of the cytoplasm, right? And accordingly we have the two terms, karyokinesis and cytokinesis. So in both the cases, either it is mitosis or it is meiosis, duita te ki hobo, duita process hobo, karyokinesis hobo or cytokinesis hobo. As far as which event takes first, it is always the karyokinesis that occurs first or simultaneously cytokinesis. It is not like this, the karyokinesis completely over hoi jabo tar pisote cytokinesis hobo tenukwa nohoy. Right? Karyokinesis or jite end almost around, if you have already read, I think you have heard the word called as telophase, that is the end stage for a nuclear division, no? By the time telophase hoi thake, simultaneously ki hoy cytokinesis o arambho hoi jay. Not necessarily like this that one process will completely be over and then the next process will start. Tenukwa nohoy. Simultaneously hobo, ketia hobo, last phase of karyokinesis, that is we are talking about telophase, right? Okay, next, before beginning mitosis and meiosis in details, again as we have said, since we have to talk about the nuclear division first. So, nuclear division or logot stages are involved. Our stages boli kole obviously precisely divided into 4 stages, that is prophase, metaphase, anaphase or telophase. Mitosis oto dekha pam, meiosis oto dekha pam. Talking about prophase, specific characteristics of prophase, eta cell prophase ot ase boli kole, you have to see certain types of characteristics. Kiba kibi hobo laibo cell to bitor, tete ha mi tak ki boli kom, it is in the prophase stage. Similarly, kiba kibi hobo laibo, then only we will say that the cell is in the metaphase stage. So, the specific characteristics of prophase, metaphase, anaphase and telophase, hekhini ami poribo laibo. That is one part of the question. Second part of the question is a diagrammatic based question. Diagram to aki dibo cellor and you have to identify whether the cell is undergoing prophase, metaphase, anaphase and telophase. Generally, maximum question has been asked about karyokinesis and lesser about cytokinesis. Cytokinesis any questions eku nai. Apart from the fact that cytokinesis in case of a plant cell and in case of an animal cell is different. Or slightly different, maximum questions regarding the stages of karyokinesis and what is the unique point about each phase, right? Meiosis, again, if you have heard, meiosis is broadly divided into 2 parts or 2 types, that is meiosis 1 and meiosis 2. Similarly, meiosis 1 or ki hobo? Karyokinesis, cytokinesis hobo. Meiosis 2 ki hobo? Tatu karyokinesis or cytokinesis hobo. Both the 2 processes will occur in both meiosis 1 and meiosis 2. So, if we say that both the process occurs in meiosis 1 and meiosis 2, then obviously the stages would be the same. Prophase meiosis oto hobo 1 or meiosis 2 to prophase hobo. Thik teneke metaphase 1 and metaphase 2, telophase, anaphase 1 and anaphase 2. Again, what you have to do is the difference between prophase 1 and prophase 2. Teneke ami comparative study eta koribo laibo. Anaphase 1 and anaphase 2. And next is, there are 3 words. Suppose, if I take anaphase as a stage, okay, that is 3 word associated with anaphase, anaphase 1 and anaphase 2. Right? If you don't precisely mention anaphase 1 or anaphase 2 boli mention no kori, okol jodi anaphase likha, it is obvious that we are talking about mitosis. If you want to write the points about meiosis, it is very much essential that you write anaphase 1, then anaphase 2. 1 ba 2 likhiboi lagibo. Jodi okol anaphase likha, we refer it to mitosis. Anaphase kot hoy? Mitosis. Anaphase 1 or 2 kot hoy? Meiosis. Clear? Similarly, prophase kot hoy? Mitosis or prophase 1 or prophase 2 kot hoy? Meiosis. And meiosis or prophase is a lengthier process. We have 5 stages of prophase. We will be studying about it later. Okay, today we will start the class with mitosis. So, heading koriba mitosis. So, I mean introduction of mitosis or basic points ki to ami gom paisu that this is a process of cell division where the mother cell will divide and form 2 daughter cells having equal number of chromosomes. Since this has equal number of chromosomes, it will be called as equational division, right? Next point about mitosis, it consists of 4 phases: prophase, metaphase, anaphase and telophase, followed by cytokinesis. So, hekhiniye hol main points about mitosis. Ami okon intro likhi lo. Mitosis bole likha okay. Mitose is equal to thread, os is equal to stage. By the word thread, what are we talking about? We are talking about thread-like nature of the chromosomes, right? Because I think you all remember the difference between chromatin network and chromosomes. Chromatin network ki hoy, the thread-like structures. Where will you see? Obviously inside the nucleus. Chromosomes ko dekha paba, that also inside the nucleus. Kintu ketia dekha paba? Before dividing and during division. Before dividing, ki dekha pam ami chromatin network. Aru as the process of division starts, ami ki dekha pam chromosomes started. somatic cells aru germ cells, no? Somatic cells mane body cell, germ cells mane reproductive cells. So, the introduction. Right. So, described by question ahise, right? Generally, Fleming or put question ahise, right? So, we have to remember this. The term mitosis was given by, even that is asked. Common method of cell division because it occurs in key body cells, no? Next is it occurs in somatic cells or body cells, we understood, also called as equational division, another important point. Equational division mane we understood, right? Next, duration of mitosis, ER2 question ahise, 30 minutes to 3 hours, and it consists of two phases, that is karyokinesis and cytokinesis, right? And if we talk about karyokinesis, cyta stage, prophase again divided into 3 stages, that is early prophase, mid prophase and late prophase. Next is আমি prophase heading কৰিম. Below prophase, okay, I'll write it here so that you can see. Actually, there was no space, so I had to write it like that. Is it okay? Is it over? তলৰখিনি বাকি আছিল So each and every phase in details we need to talk about with the proper diagram. So heading koriba prophase. Right? Okay. Okay, before going in detail, just to have a rough idea, we are talking about a cell. Cell or ki thakibo? Nucleus. Nucleus too, and this is the cytoplasm. If a cell needs to divide, then the nucleus needs to divide, cytoplasm needs to divide. So first phase of ki hobo? The nucleus will start dividing, right? When it starts dividing, first phase to kami ki boli kom? Prophase. What happens in prophase? There are certain changes that occurs in the nucleus. Key changes ase ami specifically likhi dim. Out of all the changes, no, the first change that we see, nuclear membrane to bhangi jai, right? Yate ki ase? It is called as a nucleus because it has a nuclear membrane. No, eta nucleus ok ami nucleus boli ketia ko? When it has a proper membrane. In case of prokaryotes, the prokaryotes do not have a nucleus. What is the reason behind? Because they have the genetic contents, but the contents are not surrounded by a particular membrane. So whenever it is surrounded by a membrane, we can use a word called as nucleus. So what happens? The nucleus needs to divide. What is the first step? It is degradation or breaking of the nuclear membrane, right? So it is first step. So prophase or JTI prophase kuna, this is the first line that you have to remember. Now as we have written, prophase is again divided into 3 steps, no? Early prophase, mid prophase or late prophase. So a nuclear membrane kon se bhangi se, kuntu stage of prophase of bhangi se, that is important. Does it disappear or does it start breaking down in early prophase, mid prophase or late prophase, right? So we will be talking about specific characteristics about each stages and prophase in general. Early prophase ki hoy, mid prophase ki hoy, late prophase ki hoy, kintu ami likhime. Kintu prophase as general, but prophase in general, kan prophase is something that you will hear in case of meiosis also, right? So prophase boli kole, apart from the question to jodi specifically ahise, no, je early prophase ki hoy, mid prophase ki hoy, teneke nahi pelai. Prophase ki hoy boli jodi ahe in general, prophase ki hoy, nuclear membrane kon bhangi jai. That's the first line you remember. Right? Next, so that's the first point. Second, in case of prophase, something happens to the genetic material. So yate ki asile, when we talk about a nucleus or inside jiman genetic material asile, prior to division, those were called as what? Chromatin network, right? Those were thread-like in structures. In case of prophase, ki hobo, 8 thread-like structures bilak, ami in the form of chromosomes, they will start behaving or they will start appearing. So if a chromatin network has to appear in the form of a chromosome, tetehale ki hobo leibo? Condensation of the genetic material, right? Condensation mane ki? Horu hoi jabo. Thread asile, elongated hoi gesile, tahale condensed hoi pelai. Eta structure form koribo, he structure to ami ki boli kom? Chromosomes boli kom, right? So prophase ki hol? Nuclear membrane bhangile, condensation of the genetic material or appearing of the chromosomes. Next, we can also use the word called as, as of now we understood. Okay, next is prophase aru main bostu to nucleus. Apart from nucleus, aru kiba ki thakibo no? Remember we have talked about centrioles, right? So centrioles bilakor kam ki? Spindle fibers, right? Spindriole eta cellot keita dekha paba? 1 or 2? 2, because prior to division in a particular phase the centriole already divides. Remember we have talked about the I phase, right? Interphase of Q, the duplication of centrioles takes place, right? So nucleus to usor duta centriole dekha paba. A centriole bilake gradually they will start moving to the 2 poles, right? If this is the nucleus, yate centriole asile, centriole bilakor ki hobo, spindle fibers form koribo, and during the process of prophase, ihote separate kori pelai duta poles hole jabo. Thik ase, these are the things of prophase. Next, after this ki hobo, cell to metaphase, or sorry, it will enter the metaphase. What will happen in the metaphase? Nuclear membrane has already disappeared. So now what we can see is chromosomes. Something like this, right? All genetic material. Chromosomes are inequal diagram kyo akiso, because I think you remember the structure of a chromosome. We have talked about the centromere. We have talked about chromatids, right? Eta chromosome mod ki thake, duta chromatids thake, right? So prothomote thread like asile, thread like to chromosomes convert hoy gol. Chromosomes bilak kihot asile within the nuclear membrane. Nuclear membrane kon jetia bhangi gol, they got scattered in the entire cell. Clear? So metaphase of ultimately is scattered hoi thaka chromosomes ase, ei guti chromosomes bilak they get arranged in a particular plan, equatorial plan of arrange hoi jay. Dangor jimanta chromosomes ase, yate ekhon jetia separate hoi gol, tetia hole sob scattered hoi gol. Metaphase of ki hoy at a particular plan of arrange hoi jay. So metaphase, or you can remember it as chromosomes coming in the center or the middle portion of the cell. Right? Next point about metaphase that you have to remember is, it is the best stage where you can count the number of chromosomes in a cell. Different cell, different species, different organisms or chromosomes number po beleg thake. In case of human beings it's 46, but in case of other organisms obviously it's not 46, no? But least number of chromosomes we have talked about. We have talked about the highest number of chromosomes in case of Ophioglossum, if you remember 1260 koisilo. But anyway, so what is the best stage, or how will you study as far as the number is concerned? Scattered hoi thakile, it is very difficult to study the number. So once it get aligned in a particular plan, it is the best time to study the number of chromosomes. That is another characteristic of metaphase that you have to remember, right? So after metaphase, next ahibo ki telophase. Telophase or ki hobo, that when we talk about a chromosome, no? Sorry. Okay. We talk about the anaphase. Anaphase or ki hobo, eta chromosome split koribo. Split kori pelai duta chromatids form koribo. Agote ki asile duita chromatid log lagi pelai eta chromosome hoy asile. Yate ki hobo anaphase or splitting of the centromere. That is one line. If the centromere splits, tete hale duta part separate hoy japon na jay. Centromere ki hoisile? Log lagi asile. Right? Centromere to split kore. Te etu part separate hoy japon na jay. So ultimately what happens in anaphase is eta chromosome or pra ami duta chromatids properly dekha pam. Right? So ultimately yate suppose condensed form ot asile no, sarita ase. Yate ki hobo chromatids kini dekha palu. So what exactly happens in anaphase? The centromere splits. Anaphase centromere splits is a statement that you can use only in case of anaphase and anaphase 2. Jeneke prophase or main characteristic moi ki kolu, nuclear membrane disappears, condensation of the chromosomes. Metaphase or main characteristic ki kolu, all the chromosomes get aligned themselves in the equatorial plan. These are the statements that goes for both mitosis and meiosis. Anaphase or main most important kotha ki kolu, that the centromere splits. But here what you need to remember is anaphase ami keta porhim, anaphase porhim, anaphase 1 porhim or anaphase 2 porhim, right? Centromere splits etu kot hoy okol anaphase ot hoy or anaphase 2 the hoy. In anaphase 1 the centromere does not split. Right? Ami jeta will complete mitosis and meiosis, will be comparing both the process, and at that time we will realize that no doubt in general anaphase ot ami ki koi thoi diu, that centromere splits, but centromere split kot koribo, anaphase ot koribo aru meiosis or anaphase 2 koribo. Anaphase 1 ot centromere split nokore. Hetu uporte question ahe. What is the difference between anaphase and anaphase 1? The difference between anaphase 1 and anaphase 2. The similarity between anaphase and anaphase 2. Buji paisa? Keneke hoy process to, as we go along with the chapter we will be learning about it. But what you will study? The differences. Right? In general, anaphase ot ki hoy, since we are talking from mitosis point of view, anaphase ot ki hobo centromere to split kori eta chromosome ki hole convert hoy jabo duta chromatids hole convert. Right? Next, After this, we will have the telophase. Telophase is considered as the just the opposite of prophase. And whenever we talk about opposite of prophase, jibur prophase hoisile gutekhini ulta hobo. Agote ki hoisile? Nuclear membrane kon bhagi goisile, ate ki hobo? Nuclear membrane kon form koribo. Right? Next, tate ki hoisile? Je chromosomes bila condense hoisile, yate decondense hoi jabo. Right? So ultimately telophase yate aru stage hobo. Okol eneke thakile to no hobo no. Obviously spindle fibers form hobo. Spindle fibers form korar pisot tahate eta cellot ekhini contents yale gusi jabo, ekhini contents ekhinle gusi ahibo, duta polole gusi jabo. Tar pisot ki hobo? They will develop their own membrane, and ultimately when they develop their own membrane, ami ki boli kom? Two nuclei, duta nuclei ami kod dekha pao? Telophase. Aru jeta duta nuclei hoi ase, simultaneously ki hobo? Cytokinesis hobo. Right? So actually sabole gole these are the points that we need to remember, but note to likhi diu, and what are the questions that is asked? Ami hini sam. Say.
First case of that means the chromatin network. So আমাদের condense হয় আর chromosomes form. Actually এটা break হয়. তো chromatin network কি হবো নে?
No. Tetia tahatok chromatin buli kom, kintu chromatin buli no doubt the chromosomes will divide and form two chromatin, kintu we will be not using the word chromatin. Again they will be called as chromosomes itself. Right? Actually ki hoy chromatin e hoy, kintu we don't use the word chromatin any longer, and plus cell division or jimanpur terms se we use the word chromosomes. Chromatin word to ketia use koro? Prior to division. Right?
Chromatin network ke thake.
Actually so genetic material to genetic material hoy khali hi hokot ke hoy ase condense hoy ase ne digholia hoy ase, right? Yate condense hoy ase kitu separate hoy goise, kitu agor stage to digholia hoy asile, condense one asile. Prior to division condensed hoy na thake, only prophase eta start hoy tetia condensed hoy, right? As we write the note, keneka dhorone porhim etu likhim. সেইকাৰণে ইনভিজিবল প্ৰফেজত ডিষ্টিংক্টলি আমি দেখাহে পাম ঠিক আছে ডিভিজন চোৱা ক্ৰোমাটিন টু ক্ৰোমোজোমছ অলৰেডি তুমি এছ ফেজত হৈয়ে যায় কিন্তু আমি দেখা কেতিয়া পাম প্ৰফেজত দেখা পাম এণ্ড বিকজ অফ দ্যাট ৰিজন ৱাই ইট ৱাজ কল্ড এজ ইনভিজিবল ফেজ ৰাইট
আমি যোনকেইটা ফেজটো ডিভাইড কৰোঁ G1 S and G2 তো suppose G1 ৰ কিবা এটা এক্টিভিটি হৈছে S ত বেলেগ এটা এক্টিভিটি হয় কিন্তু হেতু G1 এক্টিভিটিটো কন্টিনিউ হৈ নাযায়
so as the cell grows contents খিনি to bari goye thakibo so if you are talking about synthesis of certain proteins that amount will keep on growing kintu initial starting point to kot hoise G1 ot hoise right so that amount will keep on growing S1 G1 kintu as far as duplication is concerned no duplication of chromosomes okol S ote hoy when it goes to G2 aru further duplication no hoy hetu tenekei thaki jabo so eta samoy gutei kita phase or bhitor duplication of chromosomes kot hobo S ote hobo G1 ba G2 t nohoy প্ৰত্যেকটো ফেজত ইউনিক পইণ্টছ কিছুমান হ'ব যেনেকৈ স্পিণ্ডল ফাইবাৰ্ছ চিন্থেচিছ ৰাইট সেইটো অকল G2 তে হ'ব সি আৰু তোমাৰ এছ ফেজত কেতিয়াও নহয় ৰাইট চ' স্পেচিফিক্স Spirumi stage, S-P-I-R-E-M-E. when we have started the discussion, what we said is prophase or first characteristic key disappearing of the nuclear membrane. But now since we know that prophase has been divided into 3 stages, early, mid or late, you have to be precise that the nuclear membrane disappears at which stage of prophase. As a whole যেতিয়া prophase koba nuclear membrane kon disappear kore, kintu kuntu stage of prophase boli jodi specially question ahe, you have to read the points carefully. Kot disappear hoy? It is only in the late prophase that the nuclear membrane completely disappears. Starting point mid. So question ahe je early prophase or nuclear membrane thakibo ne na thake? Thakibo. That is the way you will read it, right? Okay. So that's the first point that is important for examination point of view. Second is this word. What is the Spirumi stage? When the genetic material appears in the form of a woolen ball, that is what is called as a Spirumi stage. So question ahe Spirumi stage ami kot dekha pao? Option of jodi early prophase asage, answer would be early prophase. Jodi option of okol prophase thake, answer would be prophase. If the options are like this, that prophase, metaphase, anaphase and telophase, then obviously the answer would be key, prophase. But if suppose specifically mentioned it's the early, mid and this, late, tetehale Spirumi stage in the early prophase. What is Spirumi? All the genetic material in the form of a woolen ball, that is what is called as Spirumi. That means distinct chromosomes ami dekhai na etialake, right? Viscosity is the viscous nature. Viscous boli kole ki kom, the gel like nature, no? Viscosity means what happens? We know there are two things, no? Karyoplasm or cytoplasm. What is cytoplasm? The contents or gel like contents within the cell, that is called as cytoplasm. What is karyoplasm? The liquid gel like substance within the nuclear membrane, that is called as karyoplasm. What will happen if the nuclear membrane disappears? The gel like substance যোনখিনি karyoplasm আছে সেইখিনি cytoplasm লগত mix হৈ যাব না যায়. So it will thicken up and that is what is called as viscosity. Clear? Chromosomes no, ইয়াৰ পৰা আমি দেখা পাম. Early prophase তো it was in the form of a woolen ball, so obviously that's Spirumi stage. Chromatin network হৈ আছে, right? গোটেইখিনি ইম্পৰ্টেণ্ট after coiling or spiralization, spiralisation. Dehydration. एक मिनट लिखी लुआ. Degradation to hobo, degradation and disappearing or breaking down is the same meaning. Dehydration is related, dehydration meaning mane ki? Loss of water. Loss of water is a process that occurs during formation of chromosomes from the chromatin network, right? Actually we are talking like suppose if I take an example of a cloth, kapur dhui ase, you take the cloth, you put it in the bucket of water, melar agote ki kora? Sepi dia. So jetia sepi dia, teta suti hoi jay na jay. Ene dikol hoi thake, sepi dile ki hobo? Suti hoi jabo. So hey je sepi dilo, spiralization, right? We can talk about it as coiling also. At that time what happens is dehydration sepi kyo diyo, pani khini excess jabole. So that's a process that occurs with chromosomes also. So during the conversion of chromatin into chromosomes, what happens? Loss of water, and that is what is called as dehydration. Question ahead, dehydration of chromosomes kot hoy? Dehydration of chromosomes prophase hoy.
Dehydration chromosomes hoy ne?
We are using the word chromosomes as a genetic material. It is sabole kole dehydration chromosomes or chromatin network hoy, right? Each chromosome,
two sister chromation.
oh, sorry, chromatids, yeah. So what, how will you read prophase? First of prophase boli kole, jedi specifically koi diye, early, mid or late, then this is the way you will read. It is generally prophase as a whole. These are the points that you have to remember. What happens in prophase? These are the things that happens in prophase. Mitosis to prophase ase, meiosis to prophase ase, mitosis to ekini hobo, meiosis to ekini hobo. Right? Specifically jetalke mention no kore, will be confined with those informations. Right? So nuclear membrane, nucleolus, Golgi bodies, all the organelles starts disappearing, right? Then condensation of chromosomes, but condensation of chromatids kot hobo, prophase of coiling and spiralization kot hobo, yat dehydration occurs. Dehydration jodi yat hoy, telophase of ki hobo, hydration, right? Yat jodi coiling hoy, tat ki hobo, uncoiling. Right, so teneke questions will kahibo. Chromosomes become thick and size decreases. What we are talking about, condensation only. Right, dikhol asile horu hoi jay. Next, each chromosome appears double and consists of two sister chromatids. Ami prothomte diagram tut keneke dekhisile enekwa, right? Etia diagram tut ki dekhisu? Duta part hoine eta chromosome asel, etia ami eta chromosome or duta chromatids clearly ami dekha paisu. And if you are talking about a diagram like this, no, right? Etu ki hoy? Centromere, etu eta aru etu eta. Ei duita kami ki boli kau? Sister chromatids, right? So sister chromatids bilak ki hoy? A part of a chromosome clearly ami kot dekha pam in prophase. And to be precise, which part of prophase? It is in the late prophase, right? So accordingly the diagram. Eta chromosomes chromatids keita thake duta, right? Chromatin network pele ki hole convert hoy, chromosomes hole convert hoy. Whenever we use the word chromosomes, it's obvious that it consists of two parts. Chromosomes or chromatin into chromosomes, chromosomes having two parts, a stage to kot hoy. Mane enekwa eta bostu actually ami kot dekhibo lagisile in that invisible phase of I phase. I phase or eta invisible phase asile nai. Asile S phase. S phase is called as invisible phase. Why is it called as invisible phase? Because we do not see this diagram. Actually, enekwa dhoroner bostu eta already hoye jay. We have talked about chromosomes and we have talked about duplication of chromosomes also, right? So, etu jedi chromosome hoy, duplication of chromosome mane etu holle nai. Chromatids bari gol, that is what is called as duplication. A stage to actually kot hoise. Prophase hoise ne S phase hoise. S phase hoise, but we could not see at that time properly, and so it was called as invisible. Properly ami kot dekha pam, it is only on the late prophase, right? So any doubts about prophase? Prophase ki hoy? First step of karyokinesis ki hoy? Ekita events hoy, right? Sailwa kiba doubts ase jodi muk khuda. If you have any confusion regarding duplication of DNA also, please ask. Yeah, please say. No. Divide kora buli thik teneke kwa nai. Actually, prophase buli kole, the starting point, the very starting moment has been described as early prophase. Proper signs, proper characteristics, proper visible features, ami gradually as the process proceeds, ami teneke dekha pam. Thik ase, kintu starting phase ote, ami hei bostute we cannot be saying that ami chromosomes dekha pam. Starting phase of chromatids je ase, chromatids or pra chromosome hole start hobo process to. Are starting phase to the ami early buli koiso, right? It has just started, right? At that time, we cannot see the chromosomes. It's only during mid prophase that the chromosomes starts appearing gradually. Our late prophase proper chromosome or junto structure hoy hetu ami dekha pao. Clear? Okay. Any more doubts? DNA duplication bujisu no last class ot. What is DNA duplication? Increasing, in short, what we can say, increasing the number of chromatids is also called as DNA duplication, right? As far as diagram is concerned, we have already drawn the diagram. Is this part over? Okay. So if this is clear, we'll go to metaphase. Heading koriba metaphase. Terms bilak monot rakhiba, Spirumi stage, spiralization, condensation, coiling, dehydration, etc., etc. Next likha metaphase. So, diagram to account properly. When you draw the diagram, what we are drawing, that the chromosomes were scattered in the entire cell. Metaphase, what is the line that you have to remember? That all the chromosomes, now they align themselves in the equatorial plane, right? At a mid portion of alignment. Next, when we talk about these chromosomes, we have already talked about the centriole moving to the two poles. Centriole function key, spindle fiber form korbo. Spindle fiber bilakor kam ki, they will pull the chromatids, right, to the two opposite poles. So obviously spindle fibers bilak kisuman spindle fibers ki hobo centromere logot lagi thakibo. If you see the diagram, ebilak ki hoy spindle fibers, right? Jimanpur yapra long structures ba long enuka fiber like structures ulaise no. Gutebilakok ami ki boli ko spindle fibers, but if you notice gutebilak spindle fibers chromatids ot logot lagi ase niki? Nai, kisumanot lagi ase, kisumanot laga nai. So that is the way you will draw the diagram, right? So from a centriole, thread-like structures arise, correct? Ki thread-like structure? Duta, eta hol astral rays, eta hol spindle fibers. What is astral rays? Those that look like a star and short, these are called as astral rays. Junbilak long, takami ki boli ko chromosomes. Sorry, spindle fibers. Spindle fibers are divided into two types, continuous or discontinuous. Continuous spindle fibers bilaka ki hoise chromosomes logot laga nai. Discontinuous kiho karne? Karon yate break eta hol no, ako epine hol. So yar pora ki ulai continuous ulabo, discontinuous ulabo, astral rays ulabo. Clear? So points पर आप लिखो. And if you are talking about mitotic spindle, what we are talking about is the spindle fibers, right? This structure looks like a spindle, and so this is what is called as mitotic spindle. Who forms the mitotic spindle? The spindle fibers. Who forms the spindle fibers? The centriole. What is the mitotic spindle made up of? It is made up of microtubules. Remember, we have talked about that. Okay, a line to say akisu eto ki hoy imaginary line showing the equator. বুঝি পাইছো ন? This term is important. Congregation. Question ahe congregation. The event called as congregation occurs in. It occurs at metaphase. No. What is congregation? Chromosomes bilak kot asile scattered in the cell. Gutekhini kole arrange hoy gol equator le. This process of movement is called as congregation. Driver of the chromosome, centromere. From the centriole, 3 types of fibers we can see: astral rays, continuous fibers or discontinuous fibers, right? We can also talk about it as chromosomal fibers or tractile fibers, continuous fibers or astral rays. Buji paiso clear? If this is over, anaphase heading koriba. Can I rub this one? Next likha anaphase. Colorful pen, I know. It does matter. থাকিব 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 I'm not drawing it down. Continuous, discontinuous সব থাকিব হা
Continuous fibers attach that means this side and this side they match together and then
yeah it originates from this continues till here, right? Again they will go from here, they'll come here, they'll go from here, they'll come here. But if at all ইয়াতে যদি এটা ক্ৰোমোজোম থাকিব discontinuous, আকৌ এটা যদি ইয়াতে ক্ৰোমোজোম থাকিব তেতিয়াহ'লে discontinuous.
same spindle fiber of one will touch the
Other.
both one
Now both of them will synthesize and ultimately they will meet at a point. That's all. So either they will meet with in the centromere or they will be a continuous fiber. Okay. Metaphase, what was the first line about metaphase that you will remember? What happens in metaphase? Equatorial plan, right? It is already porhilo. Next is after metaphase ki hobe anaphase. What is one line that happens? Out of all the characteristics, so what's the important point ki hoy anaphase on? The chromosome splits, right? Chromosome to kene ki asile? Something like this. This part and this part, when it splits, no, ihot dutar major thake kisman protein fibers june niki tahatok dhori rakhisile, those are called as interzonal fibers. When can you see the interzonal fibers? Only during anaphase and anaphase 2 when the centromere splits. Anaphase 1 ot ami interzonal fibers dekha napao, kyo dekha napao? Karon centromere to split nokore. ঠিক আছে, এতিয়া কনফিউজ হ'ব নালাগে, this is something that we'll study at the last. As of now, interzonal fibers could be seen in, interzonal fibers could be observed only during the phase of anaphase. কেতিয়া দেখা পাম? যেতিয়া centromere split কৰিব. Centromere split কেতিয়া কৰে? Anaphase ত কৰে. Clear? So ইয়াৰ ওপৰতো question আহিছে interzonal fibers ৰ. The most important point about anaphase, the centromere splits. What will happen if the centromere splits? The chromatids will get separated. Each separated chromatid is now no longer called as chromatid, but instead we use the word key, chromosome. So, eta chromosome asile, etia keita chromosome form korile? Duta, that is the meaning of the slide. Is the point understood? Chromosome number double ketia hobo? Anaphase hobo. Kot hobo? Anaphase hobo, right? So, we have talked about duplication of DNA and not duplication of chromosomes. Duplication of DNA kot hoisile? S phase or? Duplication of chromosome kot hoise? Anaphase or. Duplication kya hobo leibo chromosomes or? Why is it necessary that the chromosome needs to get duplicated? Because if it does not get duplicated, then how it's going to be shared between two nuclei? Etal chromosome thakile kar usor jabo? It has to get duplicated. So, duplication of chromosomes occurs at dash phase of cell division. Very, very important line. Right? No doubt we know je egita kami ki boli ko chromatids ko, kintu after division chromatids bilakok ami chromatids nokoi pelai ki boli ko chromosomes. So, we have started our cell with 4 chromosomes. Now, how many number of chromosomes we have? 1, 2, 3, 4, 5, 6, 7, 8. 8 chromosome hoi gol. So, it is duplication of chromosomes hol na nai. Yes. So, what happens in anaphase? Duplication of chromosomes takes place. Is it clear? Then we have the last phase, that is telophase. Itu musuh. So, what can you see in telophase? Nucleus to duta nuclei hoise, right? Duta nucleus hobo lagile, nuclear membrane kon akko form koribo lagibo, and simultaneously what we can say is starting of cytokinesis. He karone cell to shape to okman change hoise nai, right? So, this indicates the starting of cytokinesis. It is not like this that karyokinesis will get over and then only the process of cytokinesis will start. Simultaneously hoi thakibo. begins to decondense, Since we have been talking about these points from the starting of the class itself, I don't think there is any point that you did not understand. In one line, goteikhinik je sum up kora, what do you remember about telophase? It's just the opposite of prophase. Whatever characteristics we have talked about in prophase, yate ki hobo? Opposite hobo. We have not talked about the word hydration and dehydration, but since we have already written in prophase, so yate to ami gomei pam ki hobo. Tat jodi dehydration hoise, yate hydration hobo. So there are certain points we did not write it, but we know it, right? Coiling, uncoiling kot hobo, we have written it down. Condensation, decondensation kot hobo, we know. Appearing of the nuclear membrane, disappearing of the nuclear membrane kot hobo, we understood, right? So end of telophase marks the end of karyokinesis and simultaneously is followed simultaneously by cytokinesis. So that's the reason why the cell shape changes, right? So any point that you did not understand. So today what we have discussed is, we have talked about the process called as mitosis, which is a very common method of cell division that occurs in the body cells or somatic cells. It is also called as equational division. What happens during mitosis? The nucleus divides and the cytoplasm divides. Similarly, we have two terms, karyokinesis and cytokinesis. How does the process of karyokinesis occur in case of mitosis? Karyokinesis occurs by 4 phases, that is prophase, metaphase, anaphase and telophase. What happens in the respective phases is something that we have discussed with a specific diagram. How will you read for the test or for the entrance or for your boards is the characteristics obviously, and also from diagrammatic point of view the questions could be asked, right? So diagram to DDO you have to identify which phase we are talking about, right? So jikita points mark korisu hekita bhalke poriba, and in the next class what we will do is we will be talking about cytokinesis. So next heading cytokinesis kori thoi dia. I think that's all for today. Okay, thank you.